This morning, I'm really excited to introduce Dr. Jonathan Dushoff, who's a professor in the departments of biology and mathematics and statistics at McMaster University in Canada. He's a theoretical biologist with broad interest in computational statistical approaches to practical biological questions, and particular interest in how systems are shaped at large scales by smaller scale changes. And he studies many infectious diseases. He's also the co-director of the International Clinics on Infectious Disease Dynamics and Data program, which runs clinics to teach mathematical modeling expertise in conjunction with AIMS in South Africa and the South African Center for Epidemiological Modeling and Analysis. And it was in South Africa where I first met him as part of one of their clinics, and Jonathan taught really some of the stickiest concepts to me personally about statistical modeling philosophy, as well as thinking about modeling for public health purposes. He also was kind enough to show me a couple of the best hiking spots in the area where we were staying, alongside a restaurant that had definitely the hands down best fish I've ever eaten. So we're really excited to have him here today to tell us about some of his COVID-19 work that he's carried out over the past year. So I'll let him take it away.
Thanks. So I'm assuming people can see my screen, interrupt me if not. And I'm embarrassed now. I can't believe at the beginning of the hour, I forgot where I met Spencer. I mean, I remembered many of the places where I met Spencer, but not the place where we first met. So I'm going to talk a little bit about transmission intervals and coronavirus control. I recently gave a version of this talk that took 55 minutes and I went too fast and didn't finish. So this is sort of a car crash, but hopefully I'll say some interesting things. And, I'm findable on Google and available by email. So a lot of COVID modeling questions. Where's COVID going to go? What is it going to take to get rid of it? Which control methods are working, particularly when we're doing non-pharmaceutical interventions? A lot of them come down to estimating what we call, so here's an example of a controversy from last summer about what worked in Wuhan and when, and how do you take a time series of observed incidents and infer when transmission rates were higher or lower. And a lot of these come down to thinking about big R and little r, and there's been recent work trying to disentangle exactly what we mean by a reproductive number. We have case reproductive numbers. If I'm infected on Tuesday, on average, how many people am I going to infect? And is that number going down by Wednesday? Versus the now more popular instantaneous reproductive number. Some of you will have tried this with epi-estim, which is even more counterfactual than the case reproductive number. It's basically saying if I were to freeze conditions right now, what would my reproductive number be? I've been assuming, since this is the consortium and you meet constantly, that we're all on the same page, that R is the number of new cases per cases. I should say big R. And so we have these two reproductive numbers. They're interesting to think about which one you want and which one you want in the presence of perfect data versus which one you want in the presence of imperfect data with reporting delays, which are the data that we have. Both of them, as well as the link between them and exponential rates of growth, depend on these generation intervals. And there's been a lot of talk during COVID about generation intervals or about serial intervals. And I made this slide about a year ago, but I think it's still roughly true. These transmission intervals are sort of the poor relations of the disease modeling world. We want to make models and we're just sort of scraping the literature for something we can use as a generation interval. Usually we're taking somebody else's reported serial interview. The methods are often ad hoc, and often they're not considered as sources of error when we go forward. And so our lab, and I in particular, have developed a real fascination or obsession with these transmission intervals. So what's a transmission interval? Well, the simplest one, and the one that we want but can rarely measure directly, is the generation interval. How long from the time when I'm infected to the time when I infect you. So I became infected on day 3. I infected you on day 8. That's a generation interval of 5 days. Typically, we're trying to estimate generation interval distributions. So here is my smoothing of some estimates made by the WHO during the West African Ebola outbreak. And the idea here is that it's very unlikely to transmit Ebola in the first 5 days, somewhat more likely to transmit in the next 5 days, and so on. And it's the generation intervals that link, as we saw in an early slide, link observed data, including estimates of little r, to our estimates of big R. So the transmission intervals drive epidemics, and the conventional modeling approach is to think of R, the rate at which a disease spreads through a population, as an emergent quantity, as a product of something about big R, how many cases are caused per case, and something about little g, how quickly does the process of disease development and transmission occur at the individual level. This is a multiplication sign. I'm not sure if that comes through clearly. And there's a classic perspective on this, what I would call a mechanistic perspective, that I might model Ebola spreading through a population by assuming that capital R is known. So here I have a transmission kernel, and I'll talk to you more about transmission kernels soon. But the idea here is that we know this curve, and we can factor it into the number of cases per case, which is about 2 here, and a distribution across time. So the area under the blue curve integrates to 1, and that's the generation interval. If we know the strength, if we know big R, then the faster the generations are, the faster the disease is going to spread through the population. There's another perspective we can take, and which is often driven by the practical questions that we're asking, which is a phenomenological perspective, that we can't really measure big R, for example, for HIV or early in the Ebola epidemic, but we often do see strong signs of exponential growth and the rate of exponential growth. So here's data from HIV, from Ebola, from coronavirus. These data might have been massaged or accumulated to make these patterns look clearer than they really are. But the point is that early in the epidemic, we're not observing big R. We're very often observing little r. And this reverses the role of generation interval in terms of how dangerous we think things are. If what we're observing is little r, then the faster the generation interval is, the lower big R we need to explain it. So here's a really nice visualization that came out a few years ago that said, if we're observing little r, so if we think that the number of cases is going to grow about this much in one time unit, then the more generations we have, the more quickly the disease is developing inside a person, the smaller big R that we infer to explain what we've seen. And so this is often thought of as counterintuitive, and it came to my attention first very counterintuitively in the context of the Ebola epidemic, that people, well, including me, were arguing that more burial transmission, later transmission of Ebola might actually be good news, both because it provides a specific mechanism for intervention, but also because it meant that big R was lower than some of our early estimates would have indicated. So, as I said, the generation interval is the thing we like to think about, certainly as modelers, and we can think about a generation interval as, imagine again that I get infected. So how long is it going to take me to infect you? We often think about it as the sum of 2 other periods: a latent period, the time until I become infectious, and an infectious waiting time. And this is something that so often trips up modelers, is the difference between the infectious waiting time, that is to say, on average, how long does it take me to infect you once I become infected, and the infectious period itself. And the reason this trips up modelers is that in our simple SIR and SEIR models, these are the same distribution, but they're not really the same distribution. The sameness of them emerges from the simple Markovian assumptions we make when we make ordinary differential equation models. And I'm going to try not to get tangled up in it, but I'm going to say that it's actually really cool. And it's exactly analogous to the bus waiting time paradox. The bus waiting time paradox being, if buses are Markovian, they leave the terminal every 10 minutes on average with no memory of history, then that's also going to be your experience when you get to the bus stop. You're going to wait on average 10 minutes for a bus. So what's the paradox? The paradox is that if you get to the bus stop and look back in time, you're also going to see that it was 10 minutes since the last bus. And so if buses come every 10 minutes, then by definition, they come every 20 minutes. And we can talk about it after if there's time. The point is that these two distributions come out the same under the simplest assumptions, but we shouldn't fall into the trap of assuming that they're actually the same distribution. Hopefully that was more interesting than confusing. I'm aware that it's a little confusing because not taking the time to explain it, and that seems like the right choice. So we want to know, to plug into our models, the generation interval. That's both for simple simulations and for simple calculations. We typically don't observe when individuals become infected. And so we're dealing with other sorts of things that I like to generally refer to as transmission intervals. The popular thing is to call it a serial interval. Serial interval is defined as from when I became symptomatic to when you become symptomatic, assuming that I infected you. This is observable in theory, but it may be hard to define. When you're talking to somebody with COVID, how do you define the onset of symptoms? Did your throat feel scratchy? Typically, we call it a serial interval, but we're not really probing hard at when symptoms started. We're using a convenient benchmark date, which might be when the diagnosis was made, or when the sample that was eventually going to turn out positive was collected, or simply when it's written down in the book of COVID notifications. And of course, the book now lives on a computer. And there are issues here. Some cases are never symptomatic or never diagnosed. They still have generation intervals. They may not even have these other intervals. And yet we're trying to measure things like this and make inferences about things like this. And it's sensible to think that these intervals, which really represent generations of the same process, have certain relationships, but the closer you look at those relationships, the more complicated they get. And it's all very cool. So here's a little diagram that we made for rabies. And so we're mostly assuming, so there's two key times if we're talking about serial intervals and generation intervals. One is the latent period, which ends when I become infectious. The other is the incubation period, which ends when I become symptomatic. For rabies, we assume that these are almost exactly the same, and that simplifies the picture. And we've said already that a generation interval is the time from when I become sick through my latent or incubation period, and then a waiting time until I infect you. A serial interval is a generation of the same process. It's from when I become sick to the time I infect you until the time you become sick, or until the time that you get your to be returned as positive COVID test, or whatever it is. So we can think about the steps involved, and we'll come back to them. A generation interval is a latent period plus an infectious waiting period. A serial interval is a symptomatic waiting period. So how long am I symptomatic until I infect you? Plus now your incubation period rather than mine. So there's two things. One, if we're admitting the difference between infectiousness and symptomaticity. That's the difference between latent and incubation. But there's also the fact that it's a different person in some sense involved in the second half of the serial interval. Good. So how do generation intervals link little r and big R, and what have we learned about it? And then I'll talk a little bit about serial intervals, and I'm trying hard to get somewhere with this before Lauren has to leave in 7 minutes. Okay, so many of you will be familiar with the renewal equation. Here's just a general way of thinking about how disease leads to disease. And you can shoehorn almost any kind of box model into here. You may lose some of the details that you want, but you should be able to keep all of the features that we're talking about now. A thing that's missing from the renewal equation framework is delays until observation. And that's something I'm not going to have time to talk about today, which is probably good because I probably don't have very much coherent to say about it. But here the idea is that little I is the actual rate of new infections, so not directly observed. And the idea is that infections come from infections, and that at any given time, the likelihood that somebody who was infected 2 days ago or 5 days ago or 6 days ago is going to cause a new infection can be summarized in a kernel. So K of tau is our fundamental thing. I've already shown you some pictures of it. We're also imagining that K can change through time. Some of you will have already thought that I should have susceptibles here, that one of the main things that changes K through time, that changes how many infections are caused by an infection in the past, is what proportion of the population is susceptible. Other things change it as well. Various control measures change it, and they may change it proportionally, the way we think of susceptibles changing it, or they may change it and also change the shape. We often think about counterfactuals where K of tau remains fixed. So when a disease is spreading through a new population and very few people are susceptible, maybe people haven't changed their behavior in response to the disease, or maybe they have, and we want to ask the counterfactual, well, if people in Ontario keep behaving the way they're behaving now, how will the disease continue to spread? So here is, again, an assumed kernel for Ebola, and we can factor it into an effective reproduction number and a generation interval. And it's not completely obvious why we should do that. One reason to do it, which I won't get too much, is that it's a useful paradigm for thinking of control. We want to get this curve down to this curve. That's what's going to get the disease under control. Another reason we like to think about it this way is the estimation problem, right? What we want to know is K of tau comma T, but we're not going to be able to estimate it in much detail. And much, or even most of what we do when we try to parameterize disease models is analogous to trying to observe either little r or big R and trying to estimate something about the generation interval. And if you knew little r or big R and the generation interval, you can then construct K, or at least a cross-section of K for now. And the typical way to do this is the Euler-Locke equation, which is really cool. And it basically says how much, the Euler-Locke equation is the typical way to link little r and big R. It says if I know K, I could integrate K to get big R, or I can ask about discounting growth through time to solve a linear fancy equation and figure out what little r is. And so if I know G, the Euler-Locke equation is what links big R and little r. I'll mention too that the right-hand side of this equation is a distribution, and in fact, a generation interval distribution, but not the canonical one. It's the one you would expect to observe empirically when a disease is spreading through a population. I'll mention also that the Euler-Lacke equation is identical to the famous Wallinga-Lipschitz interpretation, the generating function interpretation, which is often used in practice to do these calculations. And there's also a well-known approximation, which is that if we assume either directly or indirectly that our generation intervals have a gamma distribution, then the relationship between little r and big R can be written as, I forget what I call it, a generalized exponential. I don't know why that word doesn't appear on the slide. So the idea is basically that big R is a function of the product of little r, the speed, sorry, the population level speed and the generation time. And there's a function you put it through, which is either a linear function when we have exponential distributions or an exponential function if generation intervals are fixed, and in general is something between that. And this is the exact sense in which little r is a product of something about quickness and something about strength, where little l is the inverse of capital X. And we've written stuff about this. The interpretation, as I said before, is that quicker generations mean that if we knew big R, little r would get faster as our generations get quicker. And the converse of that is if we know big R, quicker generations mean that big R can be smaller and still explain the observed little r. More variation, so kappa, I think I didn't say, is a unitless measure of how much variation we have in our generation interval distribution. And the more variation we have, that's a lot like the disease spreading quicker, because some people will spread it more quickly, and the people who get it quickly can spread it again. It's like compound interest. So we lost Lauren already. Is that true? I have no idea what's going on. And what we've shown is that these approximations work really well. So this is a fit to the Ebola model. And what you see here, the black line is the linear relationship you would expect between little r and big R if you had an ODE. The other black line is the exponential relationship you would expect if you had a fixed generation model. The blue curve is the relationship you expect from a detailed model of how Ebola spreads through a population. And the black curve in the middle is what you get from estimating just one extra parameter, which is to say kappa, which is to say the breadth of the generation interval distribution. And then the black points, which match both the blue curve and the black curve in the middle, are detailed estimates for the 3 countries that had the Ebola outbreak. And we've shown that this generalized exponential is a simple scaffold on which you can evaluate estimates of big R and compare them and propagate error. Wow, I knew I wasn't going to get through it, but I kind of thought I was going to do better than this. So I'll just say, I'm going to skip the last section of my talk and try to tie up this section. So one of the things we've done trying to think about generation intervals is that there's more than one, even before we get to other observation intervals, there's more than one generation interval. There's the one modelers like to put in, which is to say, how infectious am I at a given time after being infected? There's the forward interval, which is the one that drives the epidemic forward. When will the people infected today infect others? And we might expect these two to be similar, or we might ask when they would be different. And I'll spoil it by saying they're going to be different if the number of susceptibles is going down during the period when I'm infectious, then the forward interval is going to be short compared to the intrinsic interval. We could also ask who's been infected today or who's been infected up until now, and when were they infected? And that's not going to match, that's what we measure. That's not going to match the intrinsic interval, which is what we assume, and it's not going to match the forward interval, which is what we want in order to do our calculations. And the reason for that is that when I was infected depends not only on how infectious the people infected 3 days ago are, but also how many of them there are. And so in particular, if I was infected with HIV in 1994 in Southern Africa, I could say, well, when did the person who infected me get infected? And the answer is probably 1991 or 1992, not because HIV has generation intervals that are that short, it doesn't, but because before 1990, there was basically nobody infected. The person who infected me wasn't infected in 1989, not because if they had been, they wouldn't be, but because nobody in my country was infected in 1989. And this is a very real effect, which makes measured generation intervals shorter than the generation intervals we measure are shorter than the ones we need to calibrate our models. And we can correct for this once we understand it. So that's cool. This is a stochastic simulation where the points are the realized generation intervals in a large population, and the black line is the theoretical curve. So this is a real effect that we know how to correct for. We see similar effects on networks, and they're harder to correct for. And I feel that I need to stop. I'll just say quickly that when you get into serial interval distributions, there are similar kinds of complexities and similarly cool problems, some of which we can solve in a reasonably satisfactory way, and some of which we can't. So here's all of this stuff that I might have been able to say, if not for Midas. And I will conclude by saying that strength and, and it's really time for me to conclude. Strength and speed are complementary ways of understanding epidemic growth and control. Transmission intervals are key to linking these measurements, and we need to define our transmission intervals clearly and think about how we're measuring them, how we're estimating them, and how to propagate sources of error. Thank you for the invitation and for your patience with the chaos. And as I say, I'm the only Jonathan Dushoff who's ever walked the planet. So please feel free to reach out if you have further questions, and I'll share my slides with Spencer.